In this video lecture, we're going to talk about the pH scale, acids, bases, and we'll also talk a little bit about buffers and buffering systems in our bodies. So pure water is neutral, and neutral is defined on the pH scale of equal to 7. Now, the interesting thing, though, about pH and water is that water molecules can dissociate into ions. So on the right, we have a molecule of water, H2O, and we see that this arrow is pointed both directions, which means this reaction is reversible. So a water molecule can dissociate into a hydrogen ion, okay, which is this right here, which can also be called a proton. And the other ion that will be produced is the OH minus, or the hydroxide ion. So notice we have a cation and an anion. Now, in pure water, there is very little of this dissociation, but there are exactly equal amounts of the hydrogen ion and the hydroxide ion. So that's the definition of pH 7, is we have equal amounts of those two ions. Now, the pH is actually calculated based on the concentration of the hydrogen ion in solution. So I've written here the actual equation or formula for how you would calculate pH. So if you know the concentration of the hydrogen ion in solution, you just take the negative logarithm of that. Now, it have been a while since you've had logarithms in math, but what I want to remind you about when you take the logarithm of something is it's a 10 times difference. So in other words, if there's only a change in pH of one numerical value, so if, for example, the difference between pH of 4 and pH of 5, because that's a logarithm, the actual concentration of the hydrogen ion, it's a 10 times change. So as I have written down there for you, pH of 4 means there is 1 times 10 to the negative fourth molar or moles per liter, which is equal to 0.0001 molar. pH of 5 is equal to 10 to the negative fifth molar, or 0.00001. So there's a power of 10 higher concentration of hydrogen ion in a pH 4. So notice that, let me just underline this here. 0.0001 molar, right, is 10 times less than 0.0001 molar. So as we increase the concentration of hydrogen ions, the pH values actually get lower and lower. So, do you need to be able to calculate this value? No. I just wanted to make sure that we understand that a change in one number on the pH scale is actually a 10-fold increase or decrease in the hydrogen ion concentration. Now, we've already talked that pure water is pH 7. So this is right here. This is neutral. Okay, pH 7 is neutral. Anything below 7, okay, is going to be acidic or an acid, and it gets more acidic the lower the number. Anything above 7 is going to be basic or a base. And again, the higher the number, the stronger the base. Now, we just talked about hydrogen ion concentration, and we said that pH is a measure or numerical value related to the hydrogen ion concentration. So what that means is, as the hydrogen ion concentration increases, okay, as it increases, the pH decreases. So the concentration increase is going to get lower and lower. So in other words, at pH 14 is the lowest concentration of hydrogen ion. And then as we get lower and lower towards 0, we have more and more or higher concentrations of the hydrogen ion. So everything below 7 we mentioned is an acid. This the definition of an acid that works for us is anything that increases the concentration of H plus. Because remember, the lower the acid, the higher the concentration of H plus. Our definition of a base, which is anything above 7, is going to be anything that decreases the concentration of H plus. Now, let's talk about a couple of examples. And one of these you're going to deal with a little bit in one of your virtual labs. So in your stomach has a pH of about 2. So right here on the scale where it says gastric acid, that's talking about in your stomach. And it's a strong acid that's produced there, hydrochloric acid, HCl. And sometimes we want to actually neutralize or lower the pH, raise the pH of the stomach. So we don't want the hydrogen ion concentration to be quite that concentrated. We want to raise the pH or lower the hydrogen ion concentration. And that's what antacids do. So one of your labs is going to look at the effectiveness of different antacids in neutralizing some of the pH in the stomach. Now, carbonic acid, we'll talk about that in a later slide. This is an acid that is formed in the blood because carbon dioxide gas reacts with water and forms carbonic acid. We're going to look at that as a buffer a little bit later in this talk. Now, I mentioned the term neutralization, okay? Neutralization just means when an acid and a base react and they produce water. Now, remember, pure water has a neutral pH of pH 7. So in other words, neutralization is causing this acid and this base to react to form a neutral pH that is water and then a salt. Now, I just want to make sure that I reiterate, we know that an acid is anything below 7 and it gets stronger. The acid is stronger as the number gets lower. A base is anything above 7, and again, the base is stronger as the numbers get higher. Now, hydrogen ion concentration. Is the concentration stronger in a lower pH or in a higher pH? Yes, the hydrogen ion concentration is greater at a lower pH, right? The pH is the negative logarithm of the hydrogen ion concentration. So the higher the concentration of hydrogen ion, the lower the pH value. All right, so a buffer is a combination of two things. There's a weak acid and what's called its conjugate base. And they work as partners, and the purpose for a buffer is to resist changes in pH. So in other words, if we want a stable environment like we want in our blood, then the buffer is going to resist any kind of large fluctuations in pH. Now, the way a buffer can do that is it has the ability to either accept a hydrogen ion or donate a hydrogen ion, whichever way. So in other words, they can buffer or prevent changes either way. Now, I want us to look at one that's important in our blood, and this is the carbonic acid bicarbonate combination. So the carbonic acid is the weak acid component, and the bicarbonate is the conjugate base. Now, let me show you, so focus right here on this very first chemical reaction. It looks like there's a lot going on, but I just want to simplify it for us. So I want to start with this first molecule, and that's CO2 or carbon dioxide. Carbon dioxide is a waste gas, right? We produce that in our cells and tissues, and it will be dissolved in our blood. Well, it can react with water because our blood is, right, primarily water, or a good portion of our blood is water. And when carbon dioxide and water react, they can form this acid right here, H2CO3, which this is carbonic acid. So I'm just going to draw a line right here and try to use my best handwriting. Carbonic acid. Okay. Now notice that those arrows go both ways, so that means this is a reversible reaction. Also, we can see that carbonic acid has arrows going to the right. That's also reversible. It can dissociate into a hydrogen ion and its conjugate base. So I'm going to change colors real quick and put a circle around this. Okay. This is the conjugate base bicarbonate. Okay, so we have a pair there. We have the acid, carbonic acid, and the conjugate base bicarbonate. Now, let's go back up and look. So our blood pH has a range that we need to keep our blood pH within, and that's 7.35 to 7.45. So this carbonic acid bicarbonate is going to be the buffering system that helps maintain that pH. And the way that it does is the bicarbonate piece, okay, so let me change my color here. The bicarbonate piece is the piece that's going to react with any excess acid or excess hydrogen ion. And when it does that, what it's going to produce is the other part of the buffer in the system, which is the carbonic acid. Now, the carbonic acid is present to react with any excess OH minus or the hydroxide ion. That's going to form bicarbonate, which is also part of that partner system of the buffering system. So, in other words, both components are there. If there's excess acid or excess H+, the bicarbonate reacts with it. If there's excess base or excess hydroxide ion, the carbonic acid will react with it. Now, if we just want to take a look down below, I have both examples. So let's say that we have excess base. So this right here, this hydroxide ion or anion, would represent excess base. Notice it reacts with the carbonic acid to form water and the bicarbonate. So that helps to neutralize that base. Now, what if we have excess acid? Okay, so excess acid would be represented by this hydrogen ion, right? It's going to react with the bicarbonate portion of the system. And what that produces is water and carbonic acid. So these work together in order to prevent large fluctuations in pH. Now, in your virtual lab activity, one of your activities looks at the function of buffers. And you're going to look at the capability of cytosol, which cytosol is the fluid within the cell. Now, don't confuse that with cytoplasm. They're related, but they're not exactly the same thing. Cytosol is the fluid. Cytoplasm is that cytosol plus the organelles, except the nucleus. One way that you're going to look at the capability of cytosol to be a buffer solution is with pH paper. Over to the right here, I have an image, and you can see this pH paper is based on color. So pH of 1, we have this dark pink color, and then it gets lighter pink, orange, yellow, green, all the way to very dark blue. So you would test solutions to determine their pH based on matching the color of the pH paper with this it may be a little bit different color indicator in your virtual lab, but the idea is the same. We test with pH paper, and the color tells us the pH of the solution. Now, the other component is, we've talked a little about proteins. We know that they're one of the macromolecules. We know that their three-dimensional shape is very important, and we know that if their shape is somehow altered, they become denatured, and they no longer function. So shape gets messed up, function gets messed up. So one of the things that can cause a protein to become denatured is a shift in pH. So you're looking to see what will happen to different pHs based on their ability to buffer. The next virtual lab activity, one of them looks at antacid. So we talked about stomach acid is pH of 2. And sometimes if we have an ulcer in our stomach or something going on, we might want to try to neutralize some of that stomach acid. And so you're going to look at the effectiveness of different antacids to see which ones lower the pH better. Now, instead of using pH paper, in this case, you're using something called phenol red. Okay, so just like the pH paper is an indicator, so you're looking at a color change to tell you the pH, phenol red is an indicator. And the way phenol red works, if it is in something acidic, below 7, it's going to be yellow. But if the conditions are basic or above 7, then it's going to be red. Now, I have this little pool testing kit right here. I just wanted to point out, you can't see it very well, but on the right, as part of this pool testing kit, it's testing the pH of the water, and it's using phenol red to do that. So you can see that once you add the phenol red, it has a scale here that goes from yellow to sort of reddish pink. And that's going to tell pool owners what the pH is of their pool. So in case any of you were ever lifeguards and had to test pool water, or if you have a pool at home and you've done this, then you have already experienced how phenol red works.